Hi, my name is Naomi and I'm an education ranger at Greater Sydney Parklands. I'd like to acknowledge that I am filming on Aboriginal land on Bedegal country. Today, for our virtual event From Swamp to Scrub, I'm joined by Michelle, who is a research scientist from Macquarie University.
Hi, Naomi. Thanks for introducing me, and hello, everyone out there. So, as Naomi said, my name's Michelle Power. I prefer to be just me, but I will use the pronoun she, her, and I'm on Gadigal land, and that's of the Eora Nation.
Terrific. I'm going to start by asking, what is your research about?
Well, Naomi, the research is very diverse, but the primary question that we're asking is how are parasites and bacteria transmitted between wildlife species that live in cities and people?
What type of animals would you study?
So we're looking at lots of different animals. If we're talking about what's happening in the city and animals that we're focusing on there, we're looking at possums. We're also interested in bats or flying foxes. Our work extends to areas where people have close interactions with wildlife. So, for example, conservation programs. So we're looking at Tasmanian devils, and we're also looking at sea lions and penguins. So really our work extends from animals on land to animals in the sea.
So let's talk a little bit about bats because they're just really cool animals. Why are they important to the ecosystem and why should we protect them?
So bats are working pretty hard at the moment, re-establishing burnt out forests through pollination. So they have a primary role. If you ever see a bat fly into a tree, what they're actually doing is sticking their snouts into the flowers. They're getting covered in pollen, and then they take that pollen to another tree and continue the life cycles of our important plants. So they are a really important component of ecosystems. We know that bats carry a lot of viruses, and if those viruses make their way into people, it can cause severe disease issues, okay? What we don't know about in Australian bats is disease agents or pathogens that aren't viruses. So we don't know much about the parasites that bats carry or the bacteria that they might carry that might present a public health issue.
With your research on bats or some of the other animals, how do you study them? Do you go out into the field or do you do some lab work? How do you get all this information?
Yeah, so both actually. So first of all, we actually need to get the information or the samples from the animal that we're studying. So either the bats or the possums or the penguins, seals. And we do that in a number of ways. One, we can work with wildlife rangers. The other way is that we can actually work with teams and go and trap animals as part of our research and take samples and then release them again. And the other one is what we term opportunistic sampling. And this is where we really just collect the scat sample from the animal that we're interested in. So we mentioned the colony at Centennial Park or St Parklands. We basically go in there, lay a tarpaulin down on the ground when the bats fly out to feed at night. And then when they return in the morning, before they nail down for the day to go to sleep, they will go to the toilet and they'll leave lots of poos on these tarpaulins, and then we come in and collect the samples. Once we have the samples from the field, we take them back to the lab, and in the lab we do different tests, usually on DNA, but lots of different tests for the organisms that we're interested in studying.
So with all these different animals, I'm assuming they would have different types of poos that they would look differently. Say, like just your cat and dog poo, that would look significantly different. Can you talk to me about some of the ways that we can identify different scats?
So usually you can go by the size and the shape. In the lab, we can do some extra tests. We can actually look down the microscope, and if we see bones like fish bones or plant material, we can work out, or it can help us determine who did the poo. But possums, for example, their poo is, and these are Play-Doh poos, they're not real poos that I'm about to pick up. Possum poos are about, the width of a 20 centimetre piece, and they're quite cylindrical and kind of stumpy. That's a brush-tail possum. If you have a ringtail possum, they're a little bit shorter and a little bit kind of fatter. And rabbits are very different. And if anyone's got a pet rabbit, you'll be used to seeing rabbit poo. Rabbit poos are quite cylindrical, like they're very circular poo samples. But it's easy. So for you folks at home that are watching us, you can look up pictures of poo. You can use Play-Doh, blue tack, get some brown modelling clay and build your own poo. And then perhaps you could develop a key, and you could actually use that key and a bit of a puzzle to work out who did what poo. So for example, the key might ask you, is the poo sample round or spherical? And if it is like this, it could be from a rabbit. Is it short and stumpy? It could be like this from a ringtail possum. Is it big and square? From a wombat, or is it long and cylindrical from a possum, brush-tailed possum?
Thank you for joining me today for our events from swamp to scrub. I hope the viewers as well have learnt a lot. So thank you.
Thanks, Naomi.